I am Leanne West, and I am Chief Engineer of Pediatric Technology here at Georgia Tech. And we are super excited you have joined us today for our Pediatric Tech Talk. And today, I'm really excited to learn more about a technology that is hopefully one day soon going to be able to help us tell whether or not a person's cancer is still in their body after tools that we currently have say it's gone. Um, so that's going to be our topic today, and I'm super excited to have Fatih with us today. And can you just introduce yourself to everybody who's with us?
Well, thanks, Dan. Thanks for the opportunity actually to talk about my work here. I'm an associate professor in School of Electrical and Computer Engineering at Georgia Tech. I earned my PhD at Stanford, specializing on miniaturized systems for sensing and detection applications. And then I moved to Harvard Medical School for my postdoctoral work, essentially to explore the applications of these systems, these miniaturized systems in biomedicine. Then I moved to Georgia Tech, about 10 years ago, and started my lab here. And if I, summarize work essentially that we are doing here at Georgia Tech, I would say we are aiming to develop nanotechnologies that can interface with biology at small scale. And by small scale, I mean at the molecular and cellular level. And there's a lot of value to that because a lot of health anomalies, diseases, they start with one bad molecule, gene, or a cell, and therefore the idea is essentially using this, focusing at this scale to introduce technologies that can help us detect the disease sooner, faster, or help develop effective therapeutic strategies essentially. And really, the work is really motivated by unmet needs in current practice in biomedicine in general.
Okay. And so those unmet needs are really that the the solutions we have don't tell us in time. People have cancer reoccur, and I think if we could catch it earlier, maybe we could help those people.
Definitely.
Any other unmet needs you're thinking out there, and what's your area of focus right now?
Yeah, I mean, in general, for the unmet needs, I mean, one of them, of course, is like COVID exposed us, and on that, like we really need these tools closer to us essentially test these. Maybe we want to be able to see, you want to be able to in control of our health to some degree, to be able to test ourselves, right? So we want these systems to be available anytime we need them and at home, in a simple format, so that we can kind of decentralize patient care in a way, or healthcare, and direct the resources effectively. That's one end of it. And on the other end is, of course, as you mentioned, like with cancer, there are some currently uncurable diseases, and this stems from the fact that like we don't understand the biology, like the basic research essentially is not, complete on those yet, and also like in also some extent that we don't have the tools in the clinic, like advanced tools currently in the clinic that will help us actually to better job, like in terms of detecting it earlier and maybe curing it better, essentially, like to improve patient outcomes.
Yeah, we're really excited
Mhm.
about your technology just to help meet those two needs. So, is there a specific disease that you are looking at right now?
Yeah. I think I would probably start with pediatric brain tumors in general, and specifically medulloblastoma is the one that we are actively working on right now. Um, so of course, pediatric brain tumors, I mean, it needs, cancer in general doesn't need any introduction, but in the case of pediatric brain tumors, I mean, this is one of the major cause of death from disease for children, essentially. In case of medulloblastoma, it's the most prevalent, essentially malignant type of this, and so in general, one of the scariest things about a cancer, be it a pediatric brain tumor or any type of cancer, is that it metastasizes, right? It spreads to other parts of the body, and that in most cases, including medulloblastoma, this actually kills the patient. And the way that this spread occurs is actually to very mechanical as well. Like it's you know it's when this tumor starts with this one cell and grows to be a tumor tissue, then it starts leaking these individual tumor cells like one by one to this bloodstream or other circulating fluids in the body. And if these cells survive, you just go ahead and then form another tumor tissue in somewhere else in the body. And this is how cancer metastasizes. So we are essentially, my lab, like one of the areas of research for us is that we developed a technology that can screen samples from patients to be able to and then can detect these tumor cells in that sample. And I'm saying can detect, and it's an important, it's a kind of it's a challenge, it's a technical challenge, because these cells are not easy to find. I mean, we knew that they existed more than 100 years now, that they were in the circulation. The reason that we still don't have them in the clinic is that we don't use them in the clinic, is that it's extremely hard to find them because they are mixed with these blood cells, billions of them, and you have one in a billion cell. That's the cell that you're looking for, and none of the technologies that's available in the clinic right now can actually find these cells for us to be used to use in the clinic. So the goal is essentially, for us to be able to develop this technology which can find these tumor cells out of those normal cells in the patient sample.
Yeah, I think that that's great. And so the benefit to what you're building then is that if you can detect these, then you can tell if the patient still has cancer cells in their body and that it's likely to come back.
Definitely. I mean, that's one of the benefits, and you know the way right now, in the case of medulloblastoma, for example, for pediatric brain tumors, there is no way to tell if after the tumor is surgically resected, if it can be resected. There's no effective way to know if there are still, this micro tumor tissue or minimal residual disease still in the body or not. And as a kind of a cautionary measure, essentially, like as a preventative measure, what they do is they just go and use this radiation and chemotherapies aggressively on these kids, so that it doesn't metastasize and they kill every other cell that's remaining in the body. So that, of course, it's of course also dangerous or has a lot of side effects for an adult, but also for a kid, it causes lifelong essentially morbidity, neuromorbidities that will, that probably that kid did not need it, right? So there was no way of knowing essentially whether that kid had it or not. So like tracking the minimal residual disease is one application.
Mhm.
Of course, then the other application is uh, that you can essentially track the status of the tumor. So essentially, it gives you a way of uh, performing biopsy from a sample that is so easily collected. I'm talking about the blood in this case, and there are, the current strategies of treating these patients. CSF is used, for example. CSF, they sample CSF, cerebrospinal fluid, and of course that's done through an invasive process called spinal tap, and you cannot do it all the time, and it's an invasive process. So they try to find these cells in the CSF fluid, and even those technologies are not sensitive enough. We are seeing that they are missing a lot of these cells, but in the blood they don't even look for it, and so given that you can withdraw blood samples even from a kid, like quite often, you can actually sample these tumor cells. And it's important because you can first track if the tumor is still there, as you said, but also you can sequence these cells. You can understand what is the active gene now that's driving the disease after actually, the first line of therapeutics. So you can actually tailor, continuously change and adapt your therapy to get the best chance of essential survival for this disease.
All right. So tell us about your technology and a little bit how it works so people can kind of envision what you're talking about when you're trying to identify these cells in the blood.
Yeah, so the challenge, of course, with these technologies is, and the reason that we are doing this, and the reason that it works while the current technologies fail, like in the lab, the standard laboratory technologies fail, is because of this rarity. Most of it is because of this rarity. When you have a sample, let's say in a tube of blood, you have about 50 billion cells, right? And as I said, the vast majority of this is normal blood cells, and then you have a handful of tumor cells mixed in it. So the laboratory technologies, standard laboratory practices, they use this what I call stochastic technologies, where that you perform a process and then you hope that statistically each and every cell will go to kind of the same process together, right? But it's not guaranteed. When we are using a microchip as we are building that to process a blood sample, let's say, we can make sure that each and every cell out of that 50 billion cell can be exposed to the same test, right? As they're flowing in our device, they're exposed to the same test. And I'm saying test right now because there are multiple ways that one can identify a tumor cell from blood cells, and it's essentially it's not that easy. One of the issues with the tumor cells is, of course, it's heterogeneity, right? We know that the tumor is not the same from disease to disease, from patient to patient, or even within the same patient, and it evolves constantly. So to be able to identify the tumor cells among these normal cells, you need to constantly adapt. Essentially, you have better strategies like combination of different physical mechanisms. The way that we are doing it, like the most widely used technology in my lab right now, is essentially going after the clusters or clumps of these tumor cells in circulation rather than single cells. And there's a reason for this, because most of the cancer cells that are actually released in circulation, they don't make it. It's a very ineffective process, the metastasis. Like they die on the, it's a very hostile environment for them. But if they're together, if they're traveling together with their friends, essentially, it makes them more resilient and they can survive. And that's a bad thing, because when they get out, they're still alive and they can grow to form a new tissue, new tumor tissue. So it's known for since 1950s actually, when the cells are in clusters, clumps, they are more metastatic. And so we just developed a microchip that essentially just looks for clustering or multicellularity as the cells are moving, because the blood cells don't cluster easily, and if it does, then it causes clots, other things. So that's why there are certain tricks that you can do to make sure that they don't cluster during processing. And then we just have this mechanism that we actually even turn into a simple filter-like device that you would not be able to actually differentiate from a commercial membrane filter if you buy it. And we just turn the device into this form so that you can simply just run the blood, and then the device catches the tumor cell clusters while letting the normal blood cells, single cells go. And then you can use different immunostaining standard protocols that are developed to identify these cells to detect, positively identify essentially these tumor cells.
Yes, so I think that's super interesting that those cancer cells, they cluster, and those are the ones that are bad. And
Yes,
you all have figured out a way to capture just those so that you can really identify what is still living and trying to spread in the body. I think that and like you just said, if you're capturing these clusters, then you can actually look directly at those cells to see exactly what they are, to know how to better treat the patient too.
exactly. You can, the applications time. First of all, you can just measure, like, get their concentration, right? You can just get their, morphology, how they look, like the number of them. So kind of the tumor load in bloodstream, let's say, or the circulation, and to see if, these cancer cells are coming up in numbers or going down over time, to see if the therapies are working or not. You can, as I said, you can do the molecular analysis on them to get the gene, like you can do this in a longitudinal way. Or, one exciting area is also, like, when we get them, we don't kill them. We can actually get them when they're still alive. So that gives us another opportunity kind of to take them out and do, grow them in tissue culture essentially, and start testing different drug combinations outside of the body in a cultured tissue and then see which therapies are likely to work even before administering to the patient.
Which would be a huge benefit to the patient if you're not
Definitely.
having to do these tests in general on the patient. So one of the things that just got asked in the chat is, how does the presence of clustered tumor cells correlate with clinical disease?
Well, that's the part also, like, that we are doing this in a research environment, right? And we are finding each and every day we are finding new things when you try these things, these devices on new diseases. Like in the case of medulloblastoma, for example, we're finding that these actually clusters can be used for tracking minimal residual disease, and there's nothing else available right now. So, like, we had to watch over years different patients, how they correlate with the clinical outcomes or the progression of the disease. But there are work that are published in the past decade or so showing that compared to single cells, the presence of these cells are actually the one that we need to pay attention to, to get the kind of to fight with the severity of the disease. Like if these cells start appearing, you need to essentially change your therapy or target them in a way because they are more metastatic than the single ones. So certainly in several cancers, it has been, in the research kind of stage, shown that these are the ones that one needs to really pay attention to in terms of clinical outcomes. And certainly, if there are more of them, if there are more, if there's of also a certain molecular, feature, like, they are, let's say, more mesenchymal than epithelial, then they were shown to be essentially a sign for, a kind of a negative outcome for the patient, and then you need to change your therapy.
And you can do this when other things right now say the cancer is no longer in the child. But if you do this test and you find these cells, then you need to keep going with the treatment.
Yeah, definitely.
Yeah.
I think yes. So in a way, it also helps, like, to kind of better therapy, like, to improve the therapies, because you can really personalize it and improve as you go along. Yes, definitely.
Yeah. And so I want you to give a shout out actually to your collaborator on this project. He's a clinician at Children's Healthcare of Atlanta, and maybe give some insights into why he's so interested in this and what he's been able to see clinically with some of the things you've been able to look at.
Yes. Thank you very much for actually, mentioning him. So he's my collaborator in this work. So his name is Toby McDonald, and I've been working with him now over 4 years actually in medulloblastoma, and we are seeing really exciting results. And he's essentially director of pediatric neuro-oncology in Children's Healthcare of Atlanta, and he's extremely excited about this because currently, they have essentially no way of tracking this minimal residual disease, and these preliminary results are extremely exciting because we can essentially kind of differentiate the population, which the population that's in remission versus the ones in relapse. So we are able to tell essentially which patients need this aggressive therapy versus the other ones essentially they don't need it because the disease is in remission, which is a huge thing essentially, as I said, because of these all lifelong neuromorbidities. So, like, we have a good amount of data right now that shows that this can be a very viable technology for medulloblastoma.
Wonderful. So we have another question in the chat, and it says, given cells have to cross the blood-brain barrier, are these clusters detectable in all patients with a given disease? For example, medullo versus epidymoma. So this is not my area. I'm sorry, I'm not presenting these correctly. Versus high-grade glioma, or only patients with certain molecular cytogenic features.
Sure. I mean, it's not my area as well in that sense. As I said, I'm collaborating with neuro-oncologists here, but I am familiar with those diseases now as we are working with. But the answer is essentially we focused on medulloblastoma, and I can confidently tell you that medulloblastoma, this is very high detection rate with essentially we get this in every patient that we are looking at essentially. But we also hypothesize that we will have these in other brain tumors, but we haven't checked them yet.
Okay, good. All right. And so then, are there other applications beyond just cancer and different types of cancer?
Well, yes, I over the years we applied this technology to a variety of cancers, including ovarian, prostate, breast cancers, which are all known to metastasize essentially.
Mhm.
And melanoma is another one.
Yep.
Now, this is like in the specific technology. This is for detecting tumor cells. This is one part of my work, and then the other area where we are essentially developing technologies that will help kind of develop effective immunotherapies for cancer. That's another area that we are
Mhm.
working on. And then we are also developing some essential at-home use tests. This is kind of one stream in my area, but like they're kind of related with each other. They're all about nanotechnologies for biomedical applications.
Wonderful. And I just want to tell the crowd listening, feel free to put questions in the chat. We are taking those uh, as I see them pop up. But so I love that you say this is one area. It is that kind of very sensitive detection. But why don't you tell us a little bit about some of your other work that you're doing, since you just mentioned that?
Yeah. The one area that we are related with cancer is essentially these immunotherapies. They're really giving promising results, but kind of developing those, taking one's own cells, programming it, and giving back to the body to actually fight cancer, very exciting. But doing that process is a very kind of challenging process that one needs to constantly monitor cells as they are being engineered, expanded, and one area that we are working on is essentially, again, through these microchips, but this time with the electronics built in and sensors built in, to constantly monitor them and then tell if the what we are doing is working or not, or if they are dying or not. So that's kind of one technology that we are working on, and the other one is I'm very excited about is kind of building these again microchips in a way on paper like substrate that's so that we will have this extremely simple low cost at home test that that we are all familiar now because of the pandemic. So we are kind of trying to build complex, essentially advanced tests on paper. Is another area that I'm working on.
Wonderful. I know everybody appreciates the at-home
Hmm.
tests versus having to go into a doctor's office to do it.
Yeah.
So I want to ask more on a personal question. What motivates you to do this work?
Well, so when I first started my studies, I'm an electrical engineer, so I'm very interested in small things. And when I was doing my PhD, actually, I was working on building these microscopes that are used to see individual atoms on a surface. So it was very applied physics, instrumentation heavy. I really enjoyed it. But in the end, when you build that microscope, yeah, it gets a lot of attention, and then it enables amazing basic science. But in a way, uh, it it doesn't touch directly a human's life. Like when you have a disease like cancer, for example, right? That becomes the most important problem. Puts that you put on above anything else, and it's the same applies to me. So that's why, and seeing that these devices can have this outsized effect, essentially on helping develop therapies or detecting diseases, I was really interested in that. So like then I kind of moved to a hospital environment when I started to explore essentially how these technologies can produce opportunities for meeting these needs right now in treating these incurable diseases, and that's the motivation essentially, just being able to help humans with the problem that becomes priority for everybody, or if not you, right, somebody very close by.
Yes, I think we all know someone who's been either directly or indirectly affected by cancer, and it's a horrible thing. And when it comes back after you feel like you've defeated it, that's also very defeating. And I think too, for me, when you look at a kid who has cancer, especially a brain tumor, like if you can do something to help them and get the right treatment and make it easier for them, who doesn't want to help do that, right? So,
it is really like doing work in this area is really, it's very emotional as well, because sometimes you get a blood sample that's only, that's only 0.5 milliliter, for example, right? That's very, very low amount, because that's coming from a, kid that's 6 months old, right?
yeah,
Knowing that, he or she has to fight with this disease from that, young age. Yeah, it's very emotional. Yeah.
yeah, definitely. Um, and so, thinking about that, I think you've done a lot of work with Toby over at the Children's Hospital, and I think you've actually looked at blood samples from actual patients of his to kind of see how your technology works and to see if it's able to find, these indications that cancer still may be around and compare it to, what kids do actually have it come back. Do you want to talk about that a little bit, like maybe what you all have seen with that?
Yes, so we have an amazing collaboration with Toby, the funny thing, this has started actually in the beginning. We just tested essentially this system first on a mouse model that he had, which was supposed to essentially mimic human disease. And then we just started seeing these cells, and we weren't expecting in the beginning because of the blood-brain barrier, as mentioned in one of the questions. And then then move to people like essentially kids, and we didn't only screen blood samples, we also screened the cerebrospinal fluid, like CSF samples, over a long time. Of course, CSF samples are very limited because they are collected only when medically needed, right? So the interesting thing is that most of those samples that we are getting, their analysis of their CSF samples are coming to be negative. So like there's no indication of tumor on those samples in the clinic. And also the imaging, for example, as we are monitoring it with the blood sample, we are finding these tumor cells still circulating, whereas the imaging, does not show any metastasis,
Yeah.
or clinically, there's no clinical indication. We can collect blood because the blood collection is, minimally invasive, but the imaging, follow-up imaging doesn't show anything. So, combined with that, the lack of sensitivity in this imaging and the kind of CSF cytopathology, what they're, looking at for these cells in the CSF, makes this so much valuable, because then you have a sensitivity level that's able to essentially tell you over time how the tumor load is changing, while these other technologies are showing you essentially nothing at that point, right? Even the fact that you can see these cells and monitor their level is better than what's out there, but also to be able to actually analyze it further, get the genes that needs to be essentially silenced or shut down for treatment, is a bonus that can really help improve therapies. But yes, we did this study over now 25 patients actually over years, and then we have 100% essentially detection rate. We can get, and we can, this is essentially to date the most reliable biomarker that we have for this disease. And then we are very excited actually, being able to translate this and use it in actual patient care.
Really excited about that. And I know we're at time, but there is a question in chat. So if we can maybe answer that before we hop off.
Sure.
What makes this technology applicable to children compared to adults? Is there a feature that makes it safer for children?
Well, very good question, but essentially we are working on, it's an in vitro test. So we are essentially working on collected samples, like, withdrawn blood samples. So in terms of the risk for children, there's no risk for an infection or other things, because we are working on the sample that's, out of the body essentially at that point. And this is also very applicable for adults as well. It's just that, we focused on this, the pediatric disease right now, but certainly we can expand this to adult population. But I think the kind of impact of these therapies on kids are very. I mean, there is no solution right now for this pediatric population. And Toby is an amazing, essentially, collaborator working in this area. One of the people who are at the forefront of the research here. So I think this was the right kind of location, right combination of people. This was the right essential disease to address at this point. But we are also excited to extend it to other diseases.
Wonderful. And so there is one more question. Speaking of CSF, have you collected samples simultaneously with CSF and separately as well? So between the LPs.
Between the LPs, I didn't get that part.
Oh, sorry. I think that's when you draw the CSF.
Oh, lumbar puncture.
Yeah.
Okay. So no, with the samples were collected already for clinical cytopathology. So like they just collected set sample. I think when there was an indication of either at a relapse, or a diagnosis, the clinical practice, standard clinical practice that's employed there, it require they collect CSF samples. So what we did is essentially we just had what's left from the clinical procedure there, and then that gave us a direct comparison with actually like with the sensitivity and the efficacy of the our device versus what's out there, because they were matched samples essentially. I don't know if that answered the question, but this is I wasn't involved in the collection of samples. We essentially just get whenever the clinical sample is available, we process it essentially.
Wonderful. All right. Well, I want to thank everybody for joining us today, especially you, Fatih, and all the amazing work you're doing with Dr. McDonald. I think it's super exciting. A venture capitalist recently told me he thought this was one of the most exciting things he had seen in a long time as far as cancer research goes and detection. So we really look forward to some great things coming out of this, and I really hope that this is 100% successful. So thank you so much for everything you're doing, and thank you so much to everyone who was able to join us today.
Yeah, thanks, Dan, for the opportunity. Thank you.
Absolutely. All right. Well, have a good day, and hopefully we'll see you next time at our next Tech Talk. And I don't know when that is yet, but if Christopher knows, he could put it in the chat. But thank you so much, everyone, and have a great day.
Okay. Goodbye.
Bye.